Okay, good morning students. In previous class, we have discussed regarding the chapter microbes in human welfare, and we completed that chapter microbes in human welfare. Now, in today's class, I am going to start with the new chapter, that is chapter number 11. Biotechnology, principles and processes. Biotechnology, principles and processes. Now here, one thing is to know, technology in the sense what? We are going to use some techniques, different techniques to produce something new. Okay, what we want. In the sense we are going to create some changes and we used to get the desirable product. What we expected. ನಮಗೆ ಯಾವ ಹೆಂಗೆ ಬೇಕು ಯಾವ ರೀತಿ ನಾವು ಏನು ಮಾಡಬೇಕು ಅಂತಂದಾಗ ಟೆಕ್ನಾಲಜಿ ಯೂಸ್ ಮಾಡಿ ನಮ್ಗೆ ಬೇಕಾದಂಥ ಪ್ರಾಡಕ್ಟ್ನ ಮಾಡಬೇಕು. ಅದಕ್ಕೆ ನಾವೇನು ಕರಿತೀವಿ ಮೀನ್ಸ್ ಟೆಕ್ನಾಲಜಿ ಅಂತಂದರೆ ಬಯೋಟೆಕ್ನಾಲಜಿ ಇನ್ ದ ಸೆನ್ಸ್ ವಾಟ್? ಬಯೋಟೆಕ್ನಾಲಜಿ ಇನ್ ದ ಸೆನ್ಸ್ ಹಿಯರ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ಹಿಯರ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆರ್ ಹಿಯರ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಮೇಕ್ here what is going to go for in the sense biotechnology is nothing but the deals with the techniques. Biotechnology deals with the techniques. Biotechnology is the deals with the technique by using the live organisms or the enzyme from the living organism. We used to use the whole living organism or the enzymes which are present in an organism to produce product and processes useful to human being. For using in the sense useful for the human beings, what is to use in the sense biotechnology is a branch which deals with the techniques of using the whole organism or what we are going to call as enzymes from them and produce kind of products and processes which can help the human being. That is what biotechnology. Understood? And here for biotechnology, you all study regarding the microbes in human welfare. Microbes in human welfare, what else you are studying? We are going to use the microbes for production of curd. We used to study the microbes are used for the production of beverages, vaccine. Then we used to use them as a biofertilizers. Okay. Then bioactive molecules, they are used for the production of certain organic chemicals, enzymes, etc. So what are they? They are the organisms. And in this particular chapter, what we are going to study in the sense in biotechnology, we used to use some techniques, okay, and used to get the desirable products which are developed with the help of organisms or the enzymes from the living organisms. Understood? So here what is going to happen in the sense the European Federation of Biotechnology (FEB) has given the definition for the biotechnology. They have given the definition for the biotechnology. What is the biotechnology definition? The integration of natural science and organisms, cells, part thereof. In the sense, organism, their cells, their parts. Okay, thereof, and molecular analogs for the products and services. And the molecular analogs for the products and services is called as biotechnology. Biotechnology is nothing but what? It is integration of natural science, this is combination of natural science, integration of natural science and organisms, cells, part thereof, and what we are going to call that as a molecular analogs for what products and services is called as biotechnology. In a sense, biotechnology we are going to use, it is a technique. Biotechnology deals with what? Deals with the techniques of using what we are going to call as a living organism or enzymes from the living organism to produce some kinds of products that can help for the human beings. Understood? And another thing you all know about infertility, when the couples are not getting the child, that time they used to go for the test tube baby and all. That test tube baby is also a what? One technique. Okay, used by the biotechnologist to develop or fertilize egg and ova outside the mother's means outside the organism. Understood? So in such a way that we are going to study in this particular chapter principles and processes of a biotechnology. Biotechnology it is plays a very important role to develop a kind of products which are very essential for the human beings. Okay. Here we are studying regarding the definition of biotechnology and who is given the definition of biotechnology in the sense European Federation of Biotechnology, and the definition is the integration of natural science and organisms, cells, parts thereof and molecular analogs for the products and services is called as biotechnology. Now further we will study the principles of biotechnology. What are the principles of biotechnology? There are so many principles of biotechnology. Okay, but all of means from them are two important or two core techniques that give birth to the modern biotechnology. Those are given. What we are going to call it birth to the modern biotechnology. In the sense, what are those two principles? One is genetic engineering. Genetic engineering in the sense what we are going to alter some what we are going to call that as a chemical, alter the chemistry of a genetic material. Is to alter the chemistry of a genetic material. For example, technique to alter the chemistry of a genetic material, that is DNA or RNA, DNA and RNA, to introduce into the host organism. What we used to do? We used to alter the chemistry of a genetic material DNA or RNA. In what manner we are going to alter? We used to think some kind of product further in such a way that we are going to alter the chemistry of a genetic material, DNA and RNA. Then we are going to introduce that into the host cell, into the host organism, and thus change the phenotype of organism. Phenotyping in the sense what? Phenotype in the sense external appearance. Phenotype is nothing but the external appearance. For example, I'll tell you, if you are growing the organisms for getting the meat ಅಂತಂದ್ರೆ ಮಾಂಸ ಸಂಬಂಧ ನಾವು ಏನಪ್ಪ ಅಂತಂದ್ರೆ ಆರ್ಗಾನಿಸಮ್ ಬೆಳೆಸ್ತಾ ಅಂತ ಇಟ್ಕೊಳ್ಳೋಣ. ಸಪೋಸ್ ಏನಂತಂದಾಗ ಮಸ್ಕ್ಯುಲೇಷನ್ ಬಹಳಷ್ಟು ಮಸ್ಕ್ಯುಲೇಷನ್ ಆಗಬೇಕು ಅಥವಾ ಅದೊಂದು ಸ್ವಲ್ಪ ಏನಪ್ಪ ಅಂತಂದಾಗ ಅದರ ಗ್ರೋತ್ ಫಾಸ್ಟ್ ಆಗಬೇಕಂತಂದ್ರೆ ಯಾವ ಜೀನ್ ರಿಕ್ವೈರ್ ಇರುತ್ತಿಲ್ಲ, that gene we are going to introduce into the host organism, and after that whatever the offsprings are is to come, those are altered in the sense ಅವು ಹೆಂಗೆ ಬಂದಿರ್ತಾವೆ, ನಾವು ಯಾವ ಜೀನ್ ಹೆಚ್ಚಿಗೆ ಆಗಿರ್ತೀವಿ ಆ ಜೀನ್ ಬಹಳಷ್ಟು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿ ಅದೇನಾಗಿರ್ತದೆ ಅದ್ರ ಗ್ರೋತ್ ಅರ್ಲಿ ಆಗಿರ್ತೈತಿ ಆರ್ ವೇಟ್ ಅಥವಾ ಏನಾಗಿರ್ತೈತಿ ಹೆಚ್ಚಿಗೆ ಆಗಿರ್ತೈತಿ. ದಟ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ what we are going to alter in the chemistry of a genetic material to getting desirable characters, and that we are going to write in such a way that genetic engineering principles of biotechnology genetic engineering technique to alter chemistry of genetic material DNA and RNA to introduce into the host organism and thus change the phenotype of the organism. Phenotype of the organism in the sense we are going to change the external morphology or any kinds of gene from the host by ಇದ್ದಂತ ಜೀನ್ ಅನ್ನು ನಮಗೆ ಯಾವ ಬೇಡ ಇರೋ ಅವು ಸಪ್ರೆಸ್ ಮಾಡಿ ನಮಗೆ ಯಾವ ಬೇಕಾಗಿಲ್ಲ ಅದನ್ನ ಹೋಸ್ಟ್ ಸೆಲ್ ನಾಗ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಯೂಸ್ ಟು ಗೆಟ್ ದೋಸ್ ಜೀನ್ಸ್ ಫರ್ದರ್ what we are going to call it hybridization. Understood? So hybridization ಏನು ನಾವು ಮಾಡಿದ್ದೇವೆ, we are studied about the hybridization in starting for the code enhancement ನಾವು ಗೊತ್ತಿದೆ ನಾವು ಅದರಲ್ಲ ಇದೇನಪ್ಪ ಅಂದ್ರೆ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂತ ಒಂದು ಸ್ವಲ್ಪ ಅಡ್ವಾನ್ಸ್ ಟೆಕ್ನಿಕ್ಸ್ is to use to getting the desirable characterized organisms which can help us. For example, ನೀವು ಅಲ್ಲಿ ಕಲಿತಿದ್ದು ನಾವು ಬಂದಾಗ daily management we are studied there. We studied what the hybridization and all, okay, animal breeding and all. These are all the things we have known. We were studied still also. ಅವೆಲ್ಲ ಈಗ ನಾವು ಏನು ಮಾಡಬೇಕು ಅಂತ ಅಡ್ವಾನ್ಸ್ ಟೆಕ್ನಿಕ್ಸ್ ನ ಯೂಸ್ ಮಾಡಿ we used to get the desirable characters in organism, or we are going to alter the phenotype of organism. That is what genetic engineering is. This is the first principle of biotechnology, genetic engineering. The next one is maintenance of a sterile. Here you are studying for food enhancement sterile the organism it unable to give birth to the organism. And another one, the similar meaning is become sterile in the sense it is free from any other microbes. In the sense microbial contamination free. In the sense suppose if you are going to take any petri dish or anything, if you are going to take easy ಏನಂತಂದ್ರೆ ನಾವು ಒಂದು ಪ್ಲೇಟ್ ತಗೊಂಡಿರ್ತೀವಿ ಫುಡ್ ನಾವು ಊಟ ಮಾಡೋಕೆ ಲೇಟರ್ ಆ ಪ್ಲೇಟ್ ನಾವು ವಾಶ್ ಮಾಡ್ತೀವಿ ಓಕೆ ಬಟ್ ದ ಥಿಂಗ್ ಆಫ್ಟರ್ ವಾಷಿಂಗ್ ಆಲ್ಸೋ there are many microbes okay some are useful some are harmful okay and we can't see those with the naked eyes right like that what is going to happen when we are going to develop something vaccine or something else okay that time during the development or processes for the development of that vaccine and other things okay so many what we are going to call that one as a other unwanted microbes they are going to interfere with that particular process so that time what is going to happen again agappa contamination agi ag namge yavu ಬೇಕಾಗಿರಲಿಲ್ಲ ಅದನ್ನ ಬಿಟ್ಟು ಬೇರೆದನ್ನ ಏನಾರೆ ಒಂದು ಇರ್ಬಂದಾಗ ಮೈಕ್ರೋಬ್ಸ್ ಇದ್ದಂತ ವ್ಯಾಕ್ಸಿನ್ ಬೇರೆದಂತ ಪ್ರಾಡಕ್ಟ್ ತಯಾರಾಗಿದೆ so to avoid that the biotechnology can help us to maintain the sterility in the sense microbial contamination free environment so this can be done by the what we are going to call that as a biotechnology so what else we are studied in course of in the sense we are studied regarding the biotechnology definition okay and commonly the biotechnology deals with the techniques of using the living or live organism or enzymes from the living organisms to produce product and processes useful to the human beings. And the biotechnology definition was given by the European Federation of Biotechnology. Please to remember this definition. And what are the principles in the sense? There are so many principles of biotechnology, but the main two important principles of biotechnology which can give birth to the modern biotechnology, those are one is genetic engineering, another one the biotechnology helps to maintaining or maintenance of the sterile microbial contamination free. Right? Now, next we is to study the genetic engineering. First principle what is that? Genetic engineering. Now, you all know about the what reproduction. You all know about the reproduction. There are two kinds of reproductions. What is that? Asexual and sexual. Asexual and sexual type of reproduction. And this is helpful for homozygosity in the sense they are going to maintain the similar characters from their parents to the offspring. And sexual reproduction it helps to variation. Leads to the variation. Understood? So, the sexual reproduction has advantage on asexual reproduction in the sense it provides us the offsprings in the sense of variation in a offsprings. Offsprings का ಏನಪ್ಪ ಅಂದಾಗ ಈ offsprings ಏನು ಬರ್ತಾವು parents ಇಂದ those offsprings are varies from bit to get bit variations. Understood? Here the similar kinds of offsprings we are going to get. Right? Now, how what we are going to call that one as a this in the sense in biotechnology you all know the traditional hybridization in the sense what we are going to hybridize the animals as well as plants in the sense plant breeding hybridization animal breeding these to breed the superior male and the superior female all these things but in biotechnology what we are going to go for we are going to go for creating recombinant DNA. Okay, what we are going to call that as the genetic engineering. Okay, genetic engineering it gives rise to what recombinant DNA. That is what RDNA. Recombinant DNA in the sense what earlier whatever we are studying about the animal breeding, plant breeding. Here we will discuss the traditional hybridization techniques. Traditional hybridization techniques in biotechnology are the first principle that is genetic engineering that gives us recombinant DNA. Recombinant DNA or RNA in the sense what? Recombinant in the sense, for example, I am going to draw here. This is one gene, complete organism. Okay. Now, with the help of genetic engineering, what we used to do? We used to alter the phenotype of this particular what we are going to call it a circle. For example, suppose this is the host organism. Okay. Now I used to draw another one circle. Okay. This is whatever we want in the sense if interested gene. Interested gene in the sense now we are gene that time what we are going to go for, we used to using the genetic engineering, the processes we are going to study further. Now next collectively I use in the sense then process further that here we are going to study how the genetic engineering works. Now what is going to happen in a sense, we are going to take a piece of gene from this particular what X organism, and that we are going to introduce into the host organism in the sense like this, and this we are introducing. So this is the new what we are going to call it as a offspring, or we are going to call it as a new product, that is the combination of host and the combination of desired character. Understood? In the sense, this is host DNA. In the sense, from the host DNA and from the what alien DNA and both that is called as a alien DNA. Okay, and whatever the combination is to come, that is we are going to call that as a RNA. This is the recombinant DNA. This is the recombinant DNA. In such a way that what is going to happen in the sense, this genetic engineering, it provides us recombinant DNA. In the sense, whatever we want, that gene we are going to introduce into the what we are going to call it as a host organism, and it can change the phenotype of the host organism. So, the principle that it can change the phenotype of a what we are going to call it as a host organism. So, phenotype of the host organism, this host organism, for example, this complete circle is yellow, but whatever the recombinant DNA is to come, that is the combination of yellow and green. That is what it changes the host phenotype. Okay, this is like given the example like this for understanding the concept. This is one. First one is recombinant DNA. What is that? The genetic engineering provides us the recombinant DNA. By using this, how we are going to go for the recombinant DNA? By using gene cloning and gene transfer. So here I made gene transfer. Okay. So this recombinant DNA, it is get by using the gene cloning and gene transfer. Okay. By doing gene cloning or a gene transfer in a genetic engineering, we are going to get the recombinant DNA. Understood? So what is going to happen? By doing this recombinant DNA, we are going to allow certain genes which are passed from one generation to another generation, and we used to suppress certain genes or we used to delete certain genes from the generation. In the sense, we are going to get the desirable characters or desirable phenotype in a next generation. That is the aim of what genetic engineering. That is the aim of genetic engineering. I hope you all come to know. You understand this. You understand. Understood. Next, what we are going to go for now? When we are going to study about the DNA, the DNA is going to replicate. Okay. DNA is going to replicate. See here, whatever the piece of DNA we are going to introduce, that is we are going to call that piece of DNA is alien DNA. Alien DNA. What is that? A-L-I-E-N. A-L-I-E-N. Alien DNA we are going to introduce into the host cell to getting the recombinant DNA. And this alien DNA also has capacity to replicate. Also get capacity to replicate. And in DNA we are going to get what we are going to get in the sense the DNAs, or what we are going to call it as the organisms. We are going to introduce the alien DNA into the host cell. When it gets integrated into the genome of the recipient, when it is going to integrate in a genome of a recipient, it may multiply and inherited along with the host DNA. ಅದು ಇದೇನಪ್ಪ alien DNA host DNA ದ ಹೋದಂದ್ರ host DNA ಜೊತೆ ಏನಾಗ್ತೈತಿ inherited ಆಗ್ತೈತಿ. host DNA ಜೊತೆ ಏನಾಗ್ತೈತಿ ಅಂದಾಗ inherited ಆಗ್ತೈತಿ. This is because of alien piece of DNA become a part of chromosome. Alien piece of DNA, it is going to become a part of host DNA chromosome. When we are going to introduce it to the host DNA, part of chromosome which has ability to replicate. Then this alien DNA has got ability to replicate. In a chromosome, there is a specific DNA sequence called as origin of replication. Origin of replication. O-R-I-G-I. Origin of replication in the sense, this is a DNA double stranded and it is going to go start replication, and the particular point is called as a origin of replication. You used to know the origin of replication. We are going to study that in detail further. Now we used to study whatever we are studying. These are the what basic information regarding the processes of genetic engineering. Process of genetic engineering. Now what is cloning? The term used to come here, cloning. Okay, what is meant by cloning in the sense? Cloning is nothing but making a multiple identical copies of any, making an identical copies, making an identical copies of any copies of any template DNA. Any template DNA. Any template DNA. Any template DNA. That is what we are going to call it as a cloning. And this used to come for the 1 marks. What is cloning? Cloning is making an identical copies of any template DNA. Making an identical copies of any template DNA. For example, DNA has adenine, guanine, cytosine, thymine, and they are arranged like this. In such a way, we are going to get what purines and pyrimides, purines and pyrimides on a DNA, and they are going to repeat it. For example, this is the piece. Okay, we want to get in a next generation. That time we are going to make many copies of them. That is nothing but a cloning. So, ಈ ಒಂದು piece DNA piece ಬಹಳಷ್ಟು ಕಾಪಿ ನಾವು ಮಾಡಕತ್ತಂದ್ರೆ that is what we are going to call as a cloning. In the sense, making an identical copies of any template DNA. This is complete one of template ಅಂತ ಕರೆಯುವುದು. Many template DNA. That is what we are going to call as a cloning. Okay. After this, we are going to use plasmid. We are going to use another one thing that is called as a plasmid. ಇವೆಲ್ಲ ಏನಪ್ಪ ಅಂತಂದಾಗ these all are the things which are helpful for the genetic engineering. ಈ plasmids ಒಂದು ಮಾಡಿ ನಿಮಗೆ ನಾನು next we will study the next class. Okay, what is meant by plasmid? In the sense, plasmid, it is an extra chromosomal DNA. What is that? Plasmid, it is a extra chromosomal DNA. This is circular extra chromosomal DNA, and it has self replicating capacity. Self replicating capacity. This is very important. Now, see here, plasmid. It is a circular extra chromosomal DNA and it has a self replicating capacity, and this is also we are going to use for the genetic engineering. So, still for so far in biotechnology and biotechnology principles and processes, we have studied regarding the definition of biotechnology. Okay, European Federation, that gives the European Federation of Biotechnology, that gives the definition of biotechnology. Then we have studied about the principles of biotechnology. In principles of biotechnology, we have discussed regarding the genetic engineering and maintenance of the sterility. In genetic engineering, now whatever we are studied in the sense origin of DNA replication, recombinant DNA, then we have discussed the cloning and plasmid. These all are the processes for the genetic engineering. Still there are some what we are going to call it techniques or some tools are there. Those are helpful for the what